Good morning. So let me get screen sharing going. I'm going to talk maybe for 30 minutes or so to give you an overview of the field of 21 centimeter cosmology and two places where machine learning is being actively used in 21 centimeter cosmology research. And then Dr. Chowdhury will show uh, a workbook and work you through an example of one of these applications. And I'll pause a couple times for questions. As I said, I hope this isn't going to be too long a presentation, but certainly post questions in Q&A, and we will happily address anything that comes up. So the outline of what I want to talk about, and for the next few minutes, is the field of 21 centimeter cosmology, what it is, what we're trying to measure, the science we're trying to do, and then identify, I think, the two areas in this field where AI is most actively being used. Certainly not to say the only two areas, but I think the two that have seen the deepest engagement from AI researchers. So 21 centimeter cosmology, I'm going to break this phrase up into two parts. I'm going to start by talking about cosmology, and then we'll get to the 21 centimeter part. And cosmology, I think, stands in contrast a little bit to high energy particle physics, which I think if you've been participating in some of the workshops earlier this week, you would have seen some applications of AI to particle physics. Particle physics right now is described by the Standard Model, this incredibly successful model of fundamental particles, how they make up all the constituents of matter. This has been tested in incredible detail with incredible precision at particle colliders. The Standard Model is an exceptional theory that provides the backbone of particle physics. In cosmology, we also refer to a standard model. It's a little bit fuzzier. So in cosmology, we think our universe consists of three main components. Ordinary matter is everything described by the particle physics standard model. Less than 5% of our universe is actually described by everything that we've tested in particle physics. The remainder is dark matter and the even more mysterious dark energy. And these are observed through their effects of gravity, their effects on the expansion of the universe. They're well established that we can't describe those observations with just ordinary matter, but we don't know what dark matter is and we don't know what dark energy is. Those are some of the major questions in cosmology right now, is understanding the nature of the constituents of our universe. Cosmology is also very closely related with astrophysics. Our universe today is filled with galaxies. Those galaxies are made up of stars and gas. How did they get to be the way they are? And this video shows a simulation of what's called the epoch of reionization. This is early in cosmic history, maybe 500 million years after the Big Bang. What we're seeing is the black material is neutral gas. The universe was cool, didn't have any stars in it, didn't have any galaxies in it, but they form slowly over time, growing bigger and bigger, and they put out ionizing radiation. And so what we're seeing in this movie is the neutral gas, the black gas, being eaten away, being ionized by the ever-growing galaxies and the stars within those galaxies, putting out this radiation field. And this is effectively a phase transition in which the universe goes from being neutral to, here we see at the end of this movie, ionized. What's left, those little yellow dots, are the galaxies themselves. And so we have this broad brush picture of a universe in which galaxies are forming, but we really want to understand the properties of those galaxies in detail. And so 21 centimeter cosmology, as far as the cosmology is concerned, wants to address both the fundamental physics and this astrophysics of the EOR. The 21 centimeter part of the title comes from hydrogen. So hydrogen is the most abundant element in the universe. Of that 4.9% of our universe that is ordinary matter, by mass, about 75% of that is hydrogen. And hydrogen is the simplest atom. It's one proton and one electron. But there is a very small difference between the two energy states of when the proton and the electron have their quantum mechanical spins aligned, as shown in this kind of top figure, versus anti-aligned, with one having a quantum mechanical spin up, the other down. The aligned state is slightly more energetic, and it is a very small amount of energy, about 5.9 micro electron volts. For comparison, if you've seen the Bohr model of the atom and seen the spectral lines produced by the electron changing these principal quantum energy levels, those are 1 to 10 electron volts of energy. A micro electron volt is a very small energy difference. But the atom can transition from the high energy state to the low energy state by emitting a photon that carries away that 5.9 micro electron volts of energy. And that photon, because energy and frequency are related, 5.9 times 10 to the minus 6 eV is 1420 megahertz. So it's a radio spectrum photon. In terms of wavelength, that's 21 centimeters. If you're familiar with Boltzmann statistics and can think of energy as an effective temperature, it's a very, very cold temperature, less than 0.1 Kelvin. And so all the hydrogen gas in the universe, there's enough energy, right? The universe is hotter than 0.068 Kelvin. There's enough energy that all the hydrogen gas can undergo this transition. And so these 21 centimeter photons should be produced by neutral hydrogen gas throughout the universe. And that's what we want to measure with 21 centimeter cosmology. We want to probe the universe through the hydrogen gas's 21 centimeter line. And our approach is to do this tomographically. So here's a simulation. This is a box of the universe. It's a big box. It's 1 gigaparsec on a side. That's a significant fraction of the observable universe. And this is a simulation of what that 21 centimeter signal from hydrogen would look like in this volume. And you may notice that statistically, the signal seems to change along the z-axis, that at the front of the box, the signal looks different than at the back of the box. And that's because this volume is so large, we're accounting for the fact that the light travel time from a 21 centimeter photon being emitted at the back of the box is significantly longer to us than the light travel time for a photon emitted from the front of the box. Meaning we're seeing the back of the box earlier in cosmic history. Those photons left a large fraction of the universe's history before the photons from the front of the box that are now arriving simultaneously in our detectors. And so this evolution along the line of sight is something we want to study. Basically, this z-axis lets us make that movie I showed you, where at the back of the box we see the universe when it was neutral, at the front of the box we see the universe when it was ionized. And so we want to do this with a radio telescope. I said 21 centimeter photons are radio photons. All of this was emitted significantly in the universe's past, when the universe was roughly 1/10 its current size. And so the wavelength of those photons stretches as the universe expands. So if it was 1/10 its current size, the wavelength stretches by 10. And they're now nearly 2.1 meters by the time they reach our telescope. So that's a long wavelength radio photon. So we have to build specialized radio telescopes, right? This picture is of the Murchison Wide Field Array. One element of the Murchison Wide Field Array consists of 256 tiles like these. And they're what we use to detect these 2-meter radio photons. But with these telescopes, we want to make a picture of the sky, map out that X and Y direction, and we get that Z direction from the spectrum. At the back of the box, the universe was older, the universe was smaller, excuse me, the universe was younger, it was further in cosmic past at the back of the box. It was smaller, and so that stretching of the wavelength is more significant. So when we see a 21 centimeter photon that's 2.1 meters, we know it came from when the universe was a certain fraction of its given size. When we see a 21 centimeter photon stretched to only 2 meters, we know that came from later in cosmic history. And so using the spectrum, we can get this depth of direction and actually make a tomographic map of our universe, a three-dimensional map. And that's the ultimate goal of 21 centimeter cosmology. Using the spectral line of hydrogen, get a three-dimensional distribution of all the hydrogen throughout cosmic history and understand the interplay between dark matter, dark energy, galaxy formation, and star formation. The problem is there's a lot of other stuff making 2-meter radio photons. This is a map of the Milky Way at approximately 2 meters. It's very, very bright. It's between us and all this cosmic signal we want to measure. So we call this a foreground. It's, again, in the way. And so the real challenge of 21 centimeter cosmology is how to tell the signal we want to observe apart from all of this foreground emission. The foreground emission is exceptionally bright, 4 to 5 orders of magnitude stronger than the signal we're looking for. And so separating it is very challenging. We can tell them apart from their spectral behavior. The foregrounds, we understand the physics that makes the Milky Way emit radio waves. This map I'm showing you are photons, radio photons emitted through a process called synchrotron emission. And synchrotron emission has a known spectrum. It's smooth. So from one frequency to another, the Milky Way emission doesn't change a lot. Whereas the 21 centimeter signal, each frequency we said was a different depth through our volume, it's going to change rapidly versus frequency. And so that different spectral behavior will let us separate the two signals. And that's doable. With digital signal processing techniques, we can pick out this needle in a haystack signal. It requires an exceptionally good model of the radio telescope that we're using and understanding its effects on the data we collect. But in principle, again, this is doable. There are, of course, other challenges. I'll be upfront that this idea has been around for nearly 30 years. Telescopes trying to make these observations have been around for nearly 20 years. We have not detected the 21 centimeter signal from hydrogen at cosmological distances. What are the challenges? What are stopping us? There's a number. As I said, I want to focus on two where I think machine learning and AI have really made the most contributions to date. But before I dive into this first one, seems a good moment to pause, see if there's been anything posted in the Q&A.
Not yet, but please feel free to put any questions you have.
Yes, I'll second that. So again, I'll introduce two challenges. Maybe I'll pause again after discussing this first one to see if there's been any questions. But the first thing I want to discuss is how do we identify contaminated data? Not all data we collect winds up being good data. And the majority of data contamination comes from what we call radio frequency interference, or RFI. Like people, humans, we generate a lot of radio waves with our technology. It's much, much, much brighter than the 21 centimeter signal we're trying to detect. And so if a human-generated radio signal gets mixed in with the data, that data is effectively unusable. We want to identify those contaminated samples and toss them out at the very, very beginning of our analysis. We don't want to waste computational resources processing lots and lots of data if there was bad data in there from the get-go. And when I started working in this field 15, 20 years ago, and as little as, I'd say 10 years ago, this was thought to be a solved problem. Right? Radio astronomy has been around a lot longer than 21 centimeter cosmology. Radio astronomers have had to deal with RFI for a while. They've developed algorithms to find it. And so here's an example. This is a paper from 2015, basically presenting RFI detection as it was thought to be solved at the time. So the top plot is what we call a dynamic spectrum. So on the y-axis is frequency, that's our spectral axis, and the x-axis is time. So we're looking how the spectrum changes versus time. And what you can see is at 146 megahertz, there is a signal that is actually getting stronger and broader as we go forward in time. Anything that is extremely narrow like this in frequency is very likely to be human-generated radio emission. And so the algorithm presented in this 2015 paper takes these dynamic spectra and without human interaction identifies the contaminated data. The purple in the bottom plot are the data that are identified as contaminated. And if you're wondering what all of these other horizontal lines are, that's contamination that's known to be introduced by the telescope itself. You can maybe see some faint horizontal lines towards the bottom of this plot. They're known to be corrupted. But the big thing at 146 megahertz is the identification of the radio frequency interference. And you can look at one of these plots and say, okay, we could have done this by hand, but a telescope like the MWA produces 8,000 of these dynamic spectra every 2 minutes. We need 1,000 plus hours of data to do our science. There's no way to go and identify radio frequency interference by hand. And so this is a good use case for machine learning. We have contaminating signals in these images. We want to use image-based analysis techniques to identify them. It's a little bit more complicated than a standard classification technique. Right? Instead of simply asking the always popular question of, is there a cat in this image? We need to know which pixels in this image are cat and which ones aren't. And so the standard approach has been to use an architecture called a U-net, where we start with our image, we regress down to a simplified representation, but then blow it back up to our predictions of where radio frequency interference in this image are. This example here is something of a double U-net because radio telescope data is actually a complex number. We have both an amplitude and a phase. And so this paper presents using both of those to generate our radio frequency interference classification. So it's a adaptation of a fairly standard image-based analysis technique, and it works pretty well. The results are similar to what those human algorithms do, but 20 times faster. And as data volume grows, that becomes quite important. However, this turns out not to be a solved problem, either for the classical algorithms or the machine learning algorithms. As we've been digging deeper and deeper into our data, we started detecting what we're now calling ultra-faint RFI. And so this video, you can see this little contaminating source moving down the side of the sky. This is a huge patch of the sky. These are degrees, by the way. So this is 30, 40 degrees across. And that little patch we see moving down is an airplane that is not emitting radio waves, but actually reflecting digital television broadcasts from below the horizon into our array. So normally the MWA is in a very remote location in Western Australia. It's there to avoid RFI. But when something like an airplane or a satellite crosses overhead, RFI can get reflected from below the horizon off the airplane into our array. This is very faint. And so we need more sophisticated techniques. We can't see this in a single dynamic spectra like we were looking at for those flagging techniques earlier. And so that's a challenge if it's simply not visible in the raw data products and we need to integrate to pick out even faint RFI signals. Now you might say it's faint, what do you care? The 21 centimeter signal is exceptionally faint. This is brighter than the 21 centimeter signal, so we do have to get rid of it. I think the biggest challenge for machine learning in RFI detection is that there is no ground truth. The real data is that we can't annotate it all by hand. There's so much of it. There is no way to get a hand annotated training set that so many algorithms would rely on. Furthermore, with this ultra faint RFI, we don't even know how to model it. We could look at the real data and we'd miss it. We could create simulated data, and we probably wouldn't quite get it right because there are some really subtle signatures that probably exist in human-generated broadcasts reflecting off an airplane that we wouldn't know how to put into our simulation because the physics of that reflection is so complicated. And so this field of AI for RFI flagging is 7, 8 years old now. There's been small breakthroughs, faster algorithms, slightly better training sets. But I think the real research going on right now is how to actually get a training set that's meaningful. And so there's work going on at Brown and the CFPU to generate a training set using real data. But it is ongoing. And so I'm going to leave that as kind of an open challenge for this branch of AI for 21 centimeter cosmology. And take a moment, see if there've been any questions.
No, not yet.
Okay. Um, so the other challenge I want to talk about, and this is more related to the work we'll be doing shortly, working through an example. This other challenge, I think, is still emerging. It's somewhat forward-looking, but let's suppose we actually get a measure of the 21 centimeter signal. We solve the RFI problem. We solve the telescope modeling problem. We get that tomographic map of the universe. What do we do with it? Recovering, a tomographic map at this fidelity would be an incredible feat, but how would we actually get cosmology out of it? How would we say this is what the first stars are doing, or this is what dark matter is doing? That's a regression task that machine learning is quite well suited for. We have a complicated signal. We don't have an analytic model for how that signal relates to the underlying parameters we want to constrain, but we can simulate it. And so we can use simulation-based inference to extract our parameters. And so the approach that's been around for 6 or 7 years as well now is to use convolutional neural networks, an image-based analysis technique, basically feed in slices of that cube as 2D images and build a model that regresses from an input stack of slices down to the parameters that we want to constrain. And just having a large training set of simulated slices lets us build a network that understands how that mapping works, even if we don't have an explicit model for it. And so the workshop exercise we'll do in a couple of minutes, we'll be looking at kind of single images, single slices from one of these cubes, and regressing on the neutral fraction. Yes, looks like there's a question.
There's a question asking to go over how you're going to combine them to make a tomographic map.
Um, so combine those slices, you mean?
Yeah.
Yeah, so the assumption here is that we have made a tomographic map. The machine learning analysis, we're going to pull individual slices out of that tomographic map. And
So by tomographic map, maybe can you define what you mean by
yes,
tomographic
okay.
map?
So
That may
by
be.
tomographic map, I basically mean this three-dimensional cube that we've not only made an image of the sky, but we've made the image of the sky with a depth direction to it. And that depth direction came from the frequency information, which was related to the time in cosmic history that photon was emitted. So as I said, this is a forward-looking idea of, let's suppose we can do the data analysis perfectly. We can get this cube, basically as shown on this slide, sitting as numbers on a disk, on a hard drive that we measured with our telescope. We still don't know how to get physics out of this measurement. And so that's where we're looking to progress here is analyze that cube. And so the simplest approaches have been not to treat it as a three-dimensional cube, but rather a stack of two-dimensional images, because image analysis is a more advanced field in some regards. And so we'll be looking at single slices and regressing on them shortly. I will point out that there has been work to do other architectures for current neural networks or 3D convolutional neural networks that actually treat the cube as a cube. The advances have been modest. As I will say when wrapping up, it seems like the limitation is less about the architecture we use and more about the training set that we have to put in. Not all parameter regression for cosmology is image-based. Dr. Chowdhury has done a lot of work on other forms of parameter regression, either looking at the global sky average spectrum or the power spectrum, which is a summary statistic of these fields. So this is only one example of how you might go about extracting signals from 21 centimeter data. But I think the biggest challenge is getting that training set, very similar to the RFI problem. How do we get a training set that is large enough, complex enough, and resembles the real data at a level that will ultimately lead to an approach working on real data? So what this image shows are the results of 3 different simulation algorithms for producing those maps of the 21 centimeter signal that I was showing. So each column is a different algorithm. Each row is a different slice through the cube that was ultimately simulated. And even though they all started out with the exact same setup, you can see each algorithm leads to different behaviors at a detailed level. Some produce kind of smoother bubbles, like over here. Some produce more raggedy bubbles. And you can't simulate this from first principles. Right? This is about star formation, the first stars having feedback on cosmic scales. So if we compare the size of a star to the size of the box we need to simulate, it's about 10 to the 17. There's no way to have 10 to the 17 by 10 to the 17 by 10 to the 17 pixels or voxels sitting on a hard disk being simulated. And so we have to do this approximately. We have to have models not of stars, but of galaxies or of clusters of galaxies, and not get a detailed understanding of what the very, very small scale physics is doing, but only what it does in aggregate. And so there's a concern that we've made these approximations. Do we have something that's going to translate to real data? And we don't know because we don't have the real data yet. But when a machine learning network trained on one type of simulation is applied to a machine learning network trained on another. So here's an example, parameter we want to recover called tau. We don't need to go into the details of what tau is, but it's something we want to know. It's something we know how to recover from these simulations. If we train on the blue data, we get the right value of tau back, right? If tau was 0.6, we get tau 0.06, we get it back. But if we train on the blue data and predict on the orange data, which again are just different algorithms for producing simulated 21 centimeter signals, either could be right, both are probably wrong at some level. They don't generalize to one another. And so work that we're doing at Brown, actually hope will be on the archive later this week or early next, is trying to address how we build a training set that we think will get the real physics right while ignoring the little details that distinguish these simulations from each other. And so again, I'm going to turn this over to Dr. Chowdhury in just a moment, and we'll do a workshop on building one of these convolutional neural networks for analyzing these images of 21 centimeter signal. But my overview to kind of wrap up, and then I'll take a few questions if there are any. We have nearly 20 years of data from this field. We've been operating telescopes for a long time now, but we don't have a detection of the signal. Systematic contaminants, including radio frequency interference, are still a major challenge for us. And so while we continue to improve on the traditional approaches that are, you know, standards in radio astronomy and now this nearly 2 decade old field, AI is ready to make an impact. We have these issues with training set uncertainties and training set incompleteness that I think take real interesting research to address, but the potential is there. So that's what I want to leave with and take any questions that may be out there.
So there's a question or a clarification. One of the attendees asks, uh, what does it mean to say that no detection has been made of the signal as we do detect galaxies in 21 centimeter?
That's a great question. So yes, the 21 centimeter line has been a workhorse of radio astronomy since its detection in the 40s. We've mapped out the Milky Way, we've mapped out nearby galaxies. Um, but at cosmological distances, where we're trying to map not individual objects, but gigaparsec sized volumes of the universe at very, very large scales, the signal has not been detected. I will add, uh, just within the last month, there was a paper that purports a detection of this signal from when the universe was half its current age. So not from the epoch of reionization, but still a very, very interesting result that we'll have to pay close attention to for teaching the field how to move forward.
The next question has to do with the difference between a machine learning prediction and something like Markov chain Monte Carlo reconstructions.
So
What are
that's a
the,
great question.
yeah.
So MCMC, Markov chain Monte Carlo, has been used as kind of the standard analysis technique for how to get physics out of a simulated signal, because again, we don't have the real signal. Most Markov chain Monte Carlo approaches have used summary statistics for their comparison. So this example here, this is a machine learning example from Dr. Chowdhury, but the summary statistic is called the power spectrum. That entire volume has been compressed into a one-dimensional line. Plotted here are a number of different power spectra. And so a Markov chain Monte Carlo simulates a box, calculates a power spectrum, compares it to the power spectrum of the data, and this statistic is lossy. It is not fully representative of the complexity you'd have in a volume like this. And so that's one shortcoming, that we don't have a way to extract all the information and use it in an MCMC approach. The other is very, very similar to the issue I described here, where MCMC only uses one simulation algorithm. And a paper that came out from a student in my group, and other results have been seen by other groups, show the same problem exists for MCMC, that if you simulate using one algorithm and fit in your MCMC using a different algorithm, the results don't make sense.
Pause for a second to ask if there are any other questions. Or if not, maybe we can transition
Great, yes. So
back
let,
to
let me
Doris.
turn over to Dr. Chowdhury, and I will get to work analyzing some images.
Yeah. Well, let me share the screen.
While Dr. Chowdhury is sharing screen, let me ask if there are issues with accessing the shared Google Drive and making a copy of the notebook, uh, post the questions in the Q&A.
Right. Do we wait for a few minutes or we'll just start right away?
I think we should start and then, we'll start slowly. And then if there are questions and people have run into problems, we'll deal with them as they come on.
Okay. So if you have access to the drive and the notebook, you'll see that there are two notebooks there. One which is solved CNN to predict neutral hydrogen fraction from images, and the other one without that solved part. So I would want all of you to make a copy of this particular notebook, which is not solved, and just check if you are able to access, like make a copy into your local folder, following the steps that were discussed earlier, but we'll go over that again in a while. So once you have a copy of it, and you're able to access the data files which are there in the module folder, we can start. So in this particular workshop, or in this particular module, what we'll be looking at is how we can predict the neutral hydrogen fraction from 21 centimeter differential temperature maps. So these differential temperature maps is how Dr. Jonathan Pobert just explained to us in a very nice slide. We've seen one movie, right, where we saw how the entire volume of our universe, like a box, was evolving from total a black box kind of a thing and transitioning into the universe we see today. So how the universe was transitioning from an entire volume of comprising just of neutral hydrogen to a completely ionized universe as we see today. So what we will be dealing with are slices from this kind of a simulation. So in this particular work, you'll see in the folder that there is a training set and a test set already defined. These are two pickle files. And in these pickle files, you have some slices which we'll be opening, and that's what we'll be training our network on. So the primary goal of this particular project would be to develop a two-dimensional convolutional neural network model, which will be able to predict the neutral hydrogen fraction given such a slice. So this is how the slice, one of the sample slices would look like. So originally we use a particular simulation called 21 centimeter fast. It's a semi-numerical, very state-of-the-art simulation that is used in this field. We use this particular simulation to generate light cones, or these maps of how the neutral hydrogen has evolved across different redshifts. And then we randomly pick up slices from such light cones. So each of these slices correspond to a particular redshift, and this corresponds to a particular timestamp. So we are not concerned about which particular redshift this slice is being drawn out from. Just for our information, these are how these slices look like. These are differential brightness temperature map. So these regions over here, the light and the dark regions, correspond to the ionized and the neutral regions in that particular slice. But this is not the ionization map. This is the brightness temperature map, which is proportional to the ionization field, along with certain other components as well. So essentially, we have a very, very high resolution simulation, which has each of these sides of this particular slice is about 2 gigaparsecs. And it's 512 cross 512 pixels. But for the sake of this particular exercise, we downsample it to a 64 cross 64 kind of coarsely gridded image. And this is how each of the samples would look like, what we will be training our CNN on. So essentially, what a CNN does is it looks for features in a particular map. So over here, what we have, we need to define some kind of a convolutional neural network model, which will be able to generate some feature maps from these sets of images that we have and predict the neutral hydrogen fraction. So corresponding to this particular slice, the neutral hydrogen fraction is about, it's 0.336. So these numbers are already provided. So we have from these simulations already labeled sets, like we have one image and the corresponding neutral hydrogen fraction. So what we do is we train a network and we try and predict the neutral hydrogen fractions from a test set which is unseen to the network at a later point. So how do we go about with this? So there are certain steps which are mandatory, like when we look at the methodology. The first thing is loading the data and pre-processing it. Then just knowing what the data consists of. Then we build the model. We get to know what the architecture is going to be about. Then we train the model and then we evaluate based on some validation set. And then we infer and we make predictions to new unseen data, and we also visualize the results. So in general, for a 2D regression kind of a problem, we will be having, we'll break it down step by step in the following cells as we go on running the code. So certain keywords that we need to familiarize ourselves with would be what is a loss function and what is an evaluation metric. So mostly for regression tasks, this is a regression kind of a task because we are trying to predict a particular number associated with each of the maps. So it's not a binary kind of a thing, like whether this belongs to a certain class or not. We want to predict what the neutral hydrogen fraction is corresponding to each of the images. So for such a regression kind of a task, we need to define their corresponding loss functions and the evaluation metric. So mostly we use mean squared error as the loss function, and you will be able, there are links provided in this particular notebook which you can go and find a very detailed description of all these metrics, which are very much very well documented in Keras. So the evaluation metrics which we'll be looking at in this particular notebook would be the R2 score. This R2 score is known as the coefficient of determination, and it measures the proportion of the variance of the dependent variable, which is XH1, neutral hydrogen fraction in this case, and from the predicted values. So this is how the R2 score formula would look like, and there is a ready-made function also available which you can just use. So R2 score is not the best possible metric, but it is one of the evaluation metrics that we can look at when we are trying to compare the predicted values from the model from the true values that we have, which we are aware of. And the mean squared error, it measures the averaged square difference between the predicted values and the true values as well. So these are the two evaluation metrics which we'll try to look at. And at the end, we will be we'll plot the true versus the predicted neutral hydrogen fraction for the test set, and see what would be these two metrics, what would be the values for these two metrics. So we'll get some kind of a scatter plot like this, where this solid black line is the absolute perfect prediction kind of a line. So if the prediction falls on it, that means we've got a perfect prediction. It's whatever we are expecting is what we've got from the model. And this is one of the sample models, one of the models that we have run, and this is the R2 score and the MSE that we've got. So at the end of this notebook, you'll have to generate something like this, and let us know what is the R2 score and the MSE that you've got from the model that you have generated, that you have trained. So let's start with the notebook. So I think the first few steps are very common. You have to make a copy, like I mentioned in the beginning of this particular session. So you need to mount your Google Drive and follow these steps.
I will say that for in many cases, if you have not attended a previous module, it will ask for your permission to link the Google Drive. Just hit OK a couple of times.
Yes. So once you proceed with that, you have only read access to this particular folder. So it's essential that you make a copy of the notebook. And then you'll be able to make changes to it.
Again, if you run into trouble, post in the Q&A. There's a note of someone pointing out that there's an import OS line that you might need to run in your local version.
We'll just give a couple of minutes to make a copy and get started. The first one is just loading all the necessary modules. But I hope like you've been able to make a copy of the notebook and you've been able to run the first cell over here, like importing all the necessary modules that are required for this particular notebook. So once you're done with that, we would want to load the training data and the test data from these files that we have in the folder. So this full path is something we've defined over here. This is from the shared drive and to this particular module. You read access is sufficient for this, so you did not copy the data files. You can just use it. These data sets are very small data sets, but they're sufficiently diverse. Like you have different neutral hydrogen fraction, which ranges between 0 and 1. And we have these coarsely gridded images, which correspond to many such different neutral hydrogen fraction. So essentially what the model would do is learn what features are there in that particular image slice and uh, learn the mapping between the neutral hydrogen fraction and the particular image. So once you've loaded the train data and the test data, you can print the columns to see whether you've loaded it correctly and plot any one of the slices. Over here, I've just put 9. There are several such slices. You can also print the shape and see how many samples are there in your test set. And you can pick any one of them and just plot and see how that particular slice looks like. So essentially, each of the pixels in this particular image, which looks like this, they correspond to a particular number, which is the differential brightness temperature. So we need to normalize or standardize the data set in a way that it will be easier for the network to learn. So that is what happens in the pre-processing step. So value of each pixel is the brightness temperature, and there's only one single channel. So what we do over here is we just dump the data that we have loaded into NumPy arrays, and we dump it into this X train and Y train. This just has the same data sets that you have loaded, just in two separate sets. One contains only the images and the other their corresponding labels or their corresponding neutral hydrogen fractions. So in normalization, we use something called min-max scaler. So this scales the values, normalizes the values, pixel values for each of these images so that it ranges between 0 and 1. So this is to make the learning process more efficient. So that is exactly what we do in this particular cell. So over here, we have an X train and a Y train, similarly X test and a Y test. So for each of these chunks of data set, what we do is we just apply this min-max scalar transform, and we transform it so that all the values in that particular image pixel wise is just some value between 0 and 1. It is just a normalization, which is a mandatory step for proceeding with this. Then we move on to setting up the CNN model. So there are a few terminologies, there are a few terms which you must understand in while we'll try to build a CNN model. There are there is something called batch size, then there are kernel size, pool size, and there are kernel initializers and activation functions and learning rate. Most of these you've already come across in the previous sessions as well, if you've attended them. But particularly for a CNN model, we would be looking at what a kernel size, a pool size, and what a filter is. So essentially, in a particular image, these kernel size are like smaller matrices that we slide over the image to look for features in that particular image. So the bigger your kernel size, the bigger features it's supposed to map out. The smaller your kernel size, the more finer features it's supposed to map out. So essentially we have we can use different sizes of kernels to look for features in our particular image. Then we can have multiple filters, and a combination of all these different kinds of filters and kernels will eventually extract all the features from a particular image slice that we have. So our particular, you can find more details about these of Keras sequential model, which we'll be using in this particular notebook, and different layers and the metrics provided in these links. They are very well documented on the Keras website as well. So in our dummy model, what we do is, as we've looked at in that initial image, are each of the samples, they are nothing but a 64 cross 64 grid, 64 cross 64 pixels, which is corresponding to one particular slice of this brightness temperature map. So that is what the input shape remains. Filters, this is what the default model that I have plugged in the values for. For defining the function, you can play around with these different kinds of filters and kernel size and pool size, so that you'll get a different model every time, and you can see which model performs better. Learning rate is also one hyperparameter which you control. By choosing learning rate appropriately, uh, you'll be able to generate a different model which learns these features in a better way. So mostly the model is quite sensitive to each or a combination of these parameters. So you can, for a start, you can start playing with these default values and then vary them one at a time and see what works best for our particular task. So we use a sequential model, and we construct a CNN. The input layer is the 64 cross 64 cross 1. Eventually, we flatten it out and we get one single number as the last output from the last layer. So over here, we add the filters, we add the convolutional layers, and there's a batch normalization and max pooling that also happens. And at the end of it, we flatten it and add the dense layer. And then we get our output neutral hydrogen fraction. So this is a customizable CNN model outline that has been provided. So now what I would suggest is you build your own CNN model by plugging in these values using this particular function. So to start with, you can obviously go for these default values that will run it all the same. And once your model is ready, you can run this particular CNN model for, say, 100 epochs. You can also choose the batch size. Usually, it can be any number. But once again, these hyperparameters, varying them will also vary the performance of your CNN model, not to a very large extent, but more or less, you can play around and see how differently it performs when you vary the batch size from, say, 2 to 32 or 64 or even larger. So once you're done with running the model, that is training the model, then we can look at how we will evaluate a particular CNN model for a regression kind of a task. So have you been able to load? I hope you've been able to load the model and load the pickle files, the training set and the test set, which are provided in the drive. If you are facing any difficulties, then please let us know.
If I can ask someone in the Q&A to just note whether they've been able to actually run this step, and also give a sense of, as you increase the batch size, what effect it has on the runtime. Dr. Chattery, do you have a sense of how long it would take to run 100 epochs? Is this
Yeah, it will hardly take, like, say,
anytime?
5 minutes. Yeah, it's a very brief time if you use the default values.
Okay. We will wait 3 minutes to let people catch up to
Yeah.
this.
Mhm.
So we'll pick
I
up
just
again at,
wanted to know if they've been able to load the test set and the training set. That
Hmm.
would be the starting point.
So if someone's been able to load the training set, let us know in the Q&A. Okay, we have at least one yes. And I know a
Oh,
few other
okay.
people were able to access and copy the notebook, so I think things are
Mhm.
working so far.
Okay. Then running it for 100 epochs is going to take just a few minutes. I can open the solved one as well.
There's a question in the chat about what a good target R2 score is.
Yeah, that's a good question. So we usually, if the value of R2 score is closer to 1, we can say it's a very good prediction. So the higher it is, that is closer it is to 1, the better your model is supposed to perform. So if you look at this particular scatter plot, here it's 0.98, and you can see that the scatter is less. It could be better. Maybe if you run it for more number of epochs, it will become better. So the way to look at it is, once we get these, once we finish the training for 100 epochs, we will be looking at the loss curves. So that is how we will be evaluating the CNN model. So these loss curves are nothing but the graphical representations of how well the model is learning during training. So we have a training loss curve and a validation loss curve, which we can plot and see how it is varying. The loss should ideally go down as the number of epochs increase. So if you've run this, but if you've finished training and you've got some whatever time, you can print the amount of time taken to run 100 epochs on this particular on Colab. Then what you can do is you can save the history. So when you save the history, this contains the loss evolution of the loss function and the other metrics. So in this particular cell, you will be able to plot the training and the validation loss curves. Yep.
So there is one other question about the concern as the training and especially the validation gets close to one, the worry about overfitting, or I guess the training gets close to one. Is is there a concern of overfitting in this case?
In this case, no, I didn't get closer to 1. That's not what something that we are going to look at right now. The R2 score we'll be looking at later. Right now, we're just looking at the loss curves and seeing whether these loss curves are going down as the number of epochs increase as the training progresses. So later, when we test it on a totally unseen data on the test set, then we will be computing the R2 score from the true versus predicted values. And we'll be getting the scatter plot. That's later. So that being closer to 1 has not much to do directly with overfitting. You can see that the model is overfitting if you monitor the loss curves, if you see that they are not behaving in a very, they're behaving in an abnormal way, or if the validation curve and the loss curve, they don't follow each other. There's some discrepancy in certain chunks uh, in that way, or it saturates or something like that. So once we have the training and the validation loss curves, if you plot them, Um, I could have run it as well. Let me run this. I'm hoping that you've reached up till this plots for looking at how the loss functions have evolved across the epochs. I'm running the same. Hopefully it'll be done in a couple of minutes. So in the model that I have run on this notebook, this is how the training and the validation loss curves look like. And this is how their the training and the validation metrics look like. So metrics, as in the metrics we had chosen were MAE and MSC. So this just monitors how the MSC and MAE is evolving across the number of epochs, which is 100 for our case. This is plotted in log scale. So it's not always necessary to plot it in log scale, but you see that it's going down. And if you run it for more number of epochs, it will probably reach a very smooth, saturated, constant kind of a loss where it has, we can say, assume that it's finished training perfectly. So what we note here is that the validation loss is above the training loss. The same thing goes here. So that is one of the things we check while ensuring that our model is not overfitted. So if it goes the other way around, then it also points to the fact that there might be overfitting in this case. So once we are done with looking at these the validation and loss curves, this is for a very brief, very short number of epochs that we've run here for the sake of running it very, very quickly. So you can increase the number of epochs, play with the batch size, and see what, how, to what extent you can let this loss function go down, and see whether that kind of a model gives you any better performance. So how do we know what this performance is all about? Over here, we print the model metrics. These model metrics are nothing but our MAE and MSC for our data set. So these are just the numbers that we've printed here. So now using this particular model, we make predictions. Predictions are being made on this test set that we had loaded in the beginning of this notebook. So this test set is unseen to the network. It has not been trained on it. And from this particular test set predictions is what we'll be getting the scatter plots, which we want you to plot. And because we are already aware of what the true values are, because we know what each of these slices correspond to, and we have got a prediction from this particular model. Like we have some original XH and some predicted XH. These predictions are from the CNN model, and this is what we already have. So what we'll do here is we will plot the true versus the predicted values of X neutral hydrogen fraction. So you see in this particular model that I've run, the R2 score is 0.94. The scatter is quite large. It's not very close on, but on this particular solid line. And the mean square error is, it's good. So once you play with these parameters and get a better kind of a better model as in the true and the predicted values are probably closer to each other. That always does not mean that the model is a better model, but yes, so if we get a higher R2 score, we will be very happy with the results. And with the training set that we have over here, it's a coarse gridded sample set of images, which is varying across all these different neutral hydrogen fraction ranging from 10% to about 80%. So we see that we are able to retrieve the neutral hydrogen fraction quite well. I mean, some of them are way off, but then we need to make the training set more representative. We need to have a larger training set for most cases, which will be well represented, and we must have a better model. So in better model, as in we can run it for a larger number of epochs, and we can choose a more specific, more better suited batch size, which will give us a much better prediction. So once you're done with this, you can save the model in your local drives, and then you can fine tune and see what combination of hyperparameters are giving you a much better model which you can work with. So are there any questions? Like, how did the training part, were you able to plot these loss curves? Maybe a couple of you could just say that, yes, we did the plotting.
Yeah, there's a question. So we have some successes, but also there's a question about fine tuning.
Hmm.
Uh, is there a way to set the fine tuning iteratively so that it does its model reruns itself if the
Yeah.
R2 scores is not what you would like?
Uh, yes, you can reiterate, and basically, it will not be. You can put a condition where if you want it to be based on the R2 score, but there are methods in which you can automate this by choosing a wide range of hyperparameters, like choose different combinations of these hyperparameters, the batch size, kernel size, number of filters, and you can run repeatedly and choose what the best performing model is from this. So there are automated packages currently available, which you can do it instead of doing it manually. So over here, well, why we suggest that you train and see which is a better fine-tuned model, it's just to give you an idea of how each of these parameters actually affect the training speed, training efficiency, and sometimes the performance, the predictions as well.
We have one other interesting question. One of the attendees, the model doesn't ever seem to predict very low neutral hydrogen fractions. So it flattens out at very low hydrogen densities. Is that something that is seen in other models, um, or is it
That's
a consequence of the section of data that gets selected? So you have a non-representative set of points.
Yes, yes, that could be the reason. But I'm more curious to know where exactly are you noticing that from this particular exercise?
I think
That
it's
would be very.
in their running of the model.
Mhm. Yes, so essentially that is one of the primary reasons that those lower neutral hydrogen fractions are not as well represented maybe, and there could be other reasons as well, because when we look at these images, differentiating between very low ionized regions and very highly ionized regions, it might just be complementary of each other, so there might be some kind of confusion between this particular edge of the training set and this particular end of the training set as well. So in the training process, I was just curious to know where that question came up from. Like, where did you observe that? That would be interesting to know from my side. One more way to look at it would be if you look at this entire test set that is provided to you and plot an histogram of all the different neutral hydrogen fractions that are present in the test set, you'll be able to see exactly what percentage of how well the test set is represented as well. Similarly, you can do this to check the distribution of neutral hydrogen fraction provided to you in this particular training set. So then you'll get an idea of how many, whether at all this particular training set and test set, they are well represented, equally represented for all these different neutral hydrogen fraction. It could be the case that in the training set, there is very less representation of these lower neutral hydrogen fractions, or it could be a complication that is arising because of some similarities from the network point of view while looking at very early stages of reionization and very end stages of reionization.
There's another question having to do with the learning rate.
Hmm.
One of the attendees set a very low learning rate and got a relatively low R2 value. Is that simply, and the thought is that a slower learning rate would result in a better score, but is it simply just not running for enough cycles to converge with that lower learning rate? Their learning rate was 5 times 10 to the minus 6, I think, if I count the zeros. 5 times 10 to the minus 5.
Yeah, that could be a possible reason. Maybe you have to train it for a very, very large number of epochs. That could also be the case. So each of these hyperparameters and their combinations, you cannot uniquely pinpoint that this is, if that's the only parameter that you've changed compared to the keeping all the other parameters fixed, then you'll be able to get an exact estimate of how much that particular variation in that parameter is affecting your results. Otherwise, if you've changed multiple things at the same time, then it's very difficult to pinpoint exactly what has caused that particular deviation. So essentially, if it's a slow learning rate, it might take a longer time. That is a larger
Yeah,
number of
apparently
epochs.
it was all the other parameters are the same, and I suspect that that is the case with 100 epochs and that low a learning rate,
Mhm.
you
Yeah.
would not get down to the minimum. A good test, since you have access to the module, continuing access, would be to set the number of iterations very large and see if you
Yes.
recover it. But of course, it will take time.
Yeah. So the end of this exercise, you would like get a particular, you'll get a true versus predicted values plot, which will be the final outcome of this particular exercise. So the better your R2 score is, or the closer these points lie to the solid black line, you can say your model has learned this particular training set in a better way. It's also interesting to look at the kernel size that we choose and the number of filters that we use. You could also play around with those numbers and see if you get a better model, because smaller kernels will probably be able to look at tiny deviations, tiny features over here. It will be interesting to know how changing that kernel size would affect learning the mapping between these images and the neutral hydrogen fraction as well. Well, I'm assuming that most of you have been able to like get till this last cell and get the scatter plots with at least the default values.
Let me ask a variant of that. Did anyone set the number of iterations or one of the other parameters such that they're still running the training? Looks like the answer is no, so everyone has gotten through.
Okay, so if you have any more questions, you can ask, and you have access to these notebooks. So You can run different combinations of these hyperparameters, and yeah, and this would be the final output image that we expect from this particular exercise. So with some R2 scores and some MSE corresponding to the test data set that we've provided in the drive. And also the corresponding loss curves would be very, very informative to look at. So as I mentioned, you need not always plot it in log scale, but you can just plot it loss versus epochs and these metrics versus epochs, but it becomes more clearer if you plot it in the log scale. And you can increase the number of epochs to 500,000. Even there is you can add a condition that if the loss reaches a particular point, it can stop training, like a stop loss kind of a function.
Is there a point for a given data set where adding more iterations does not actually improve the model?
Oh, yeah, that happens so many times. That means then we have to look at the training data set and see if we can diversify the data, we can make it more like well represented, like I keep saying, and basically make it larger. The more the training data set and more diverse the training data set, the better the learning process becomes. So in that case, sometimes with a smaller training set, we could, even if we increase the number of epochs, it doesn't matter. In fact, it might lead to overfitting in certain cases.
But are there other questions? I don't hear any. So you do have access to this notebook. The Solve notebook has a particular implementation of this, right? But is otherwise similar. So
Yeah, it's just similar. Only I filled out the values of those hyperparameters, and yeah, it'll run fine. If you look at the solved CNN one, it will run pretty well. But the takeaway point would be to vary these parameters and see whether you play around with it so that you are able to build your own very basic 2D CNN model to predict a numeric value, which is the neutral hydrogen fraction in our case, from these brightness temperature maps, coarse gridded brightness temperature maps from 21 centimeter fast simulation. So if you have any other questions, you can always ask later or drop a mail, and I'll be happy to answer.
So thank you to Dr. Chaudhuri.